Hello, everyone. It is my pleasure to introduce our paper submitted to iMeta. The title is The Human Skin Microbiome as a Reflective Mirror of Alcohol Intake. The skin is the largest organ in the human body, and it directly interacts with the external environment. There are 10 to the power of 4 to 10 to the power of 6 bacteria residing in every square centimeter of the skin, which is the skin microbiome. Over the past years, several independent studies have shown that the compositions of human skin microbial communities are associated with certain demographic characteristics, such as age, gender, skin care products, etc. However, we still don't fully understand how host and environmental factors shape the skin microbiome. The major purpose of this study is to integrate previous cohort studies data to identify and characterize major skin microbiome-associated factors. This is the quality control process of integrating 9 cohort studies together. The analysis of the accumulation curves on taxonomic groups of combined skin microbiome data showed that the number of classes, orders, families, and genera approached plateaus. The species number continued to increase with the maximum number of samples, indicating that the existing sample size is insufficient to include all species. We observed that 16 bacterial genera appear in the skin microbiome of more than 60% of individual, which are defined as core skin microbiome. Intergenus co-occurrence network analysis revealed that there are 5 major modules. We looked at the skin microbiomes from different skin site. The 4 major skin sites are sebaceous, moist, dry, and foot. It was found that dry skin sites have significantly higher alpha diversity. Next, we have investigated the relationship between top 10 core genus and 4 skin sites. For example, Cutibacterium are mostly correlated with sebaceous skin sites. From 9 published studies, we have organized and standardized 29 skin-related factors. We assessed the importance of these 29 factors. Continent and age group are strongly associated with the skin microbiome. Alcohol is also strongly associated with the skin microbiome. In addition, we found that our previously defined modules can distinguish the 3 status of alcohol intake. Next, we continued to examine the relationship between different genera and alcohol intake. To verify the relationship between top impact factors and skin microbiome, we performed our own skin microbiome cohort study. Age, alcohol are also top impact factors in our analysis. The trend of factor importance is consistent between the online dataset and our dataset. We found that individuals who drink different amount of alcohol also exhibit obviously different skin microbial patterns. Our defined skin microbial modules can effectively distinguish between human populations with frequent alcohol intake and those without, suggesting the co-occurrence-based module classification is a robust tool for functional analysis of the skin microbiome. We examined the relationship between alcohol intake and skin microbiome at the genus level. At the genus level, we found that Lawsonella and Streptococcus can be potential biomarkers for alcohol intake. Then, we continued to develop a random forest model that can predict alcohol intake status based on the skin microbiome at the species level. The model can reach high accuracy, and on the right panel, these are important features contributing to the model. We have performed another classical test, linear discriminate analysis effects, to identify biomarkers of alcohol intake status. Taking the overlapped biomarkers from both analyses, we found several species such as Dermacoccus nichinomiensis, Lactococcus lactis, Bravibacterium aurantiacum can be potential biomarkers of high alcohol intake. In conclusion, we have collected data from online and found that lifestyle habits are strongly associated with the human skin microbiome. To verify this finding, we have performed our own cohort study. We find that the results from the local cohort study and the online dataset are consistent. Alcohol intake is a key factor to the skin microbiome, and the skin microbiome can be a potential predictor of alcohol intake status.